Good morning, children. Welcome back to Biology class, Chapter 2, Part 7, Sexual Reproduction in Flowering Plants. Last class, we have discussed about the pollination. What is pollination? Transfer of pollen grains from anther to the stigma of a same flower or different flower of a same plant or different plant flowers with the help of wind, water, insects, bats, animals or birds. Today we are going to discuss about one new topic, pollen pistil interaction. Okay. Transfer of pollen grains from anther to the stigma. Okay, that is called pollination. But there is no guarantee right type of pollen. That is the compatible pollen. Right type of pollen of the same species should reach to the stigma. It may be wrong type either from other species or from same plant. It is called self-incompatible. Also land on the stigma. Okay. Pollens are transferring through wind or insects in land plants. You know that. Different types of pollens will reach in the stigma, and also the biotic agents will visit different types of flowers one after the other. Flowers, it may be same type of flowers or different types of flowers. That time the wrong pollens also will reach in the stigma. Wrong type means incompatible pollen grains also will reach in the stigma through these agents. Then what will happen in the next? If it is a wrong type, what will happen? Pistil has the ability to recognize the pollen whether it is compatible, that means right type, or the wrong type, incompatible pollen. See that one? Pistil has the ability to recognize the correct type or the wrong type of the pollen grains. Then if it is the right type, the pistil accepts the pollen and promotes post-pollination events that leads to fertilization. If the pollen is of wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grains and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. Okay. As mentioned earlier, following compatible pollination, correct type of the pollen grains reach on the stigma. The pollen grain germinates on the stigma to produce a pollen tube. Through one of the germ pores, the contents of the pollen grain move into the pollen tube Pollen tube grows through the tissues of the stigma and style and reaches the ovary. You would recall that some plants pollen grains are shed at a two-celled condition. What are the two cells? Vegetative cell and generative cell. In such plants, the generative cell divides and forms the two male gametes during the growth of pollen tube in the stigma. See that one? The generative cell divides and forms the male gametes during the pollen tube growth in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. You learned that last class. See that one? Then the male gametes leading to the ovary enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part if the synergids guides the entry of pollen tube. All these events, what are the events? From pollen deposition on the stigma until the pollen tubes enter the ovule are together referred to as pollen pistil interaction. Okay, this is called pollen pistil interaction. What is that? The pollen deposition on the stigma until the pollen tubes enter the ovule are together referred to as a pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating the pollen pistil interaction even in incompatible pollination to get desired hybrids. Okay. You can easily study pollen germination by dusting some pollen from flowers, some pollen from the Vinca rosea flower on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grains. Okay, that in the practicals again we can discuss. Now, to increase the crop, better quality and quantity, a plant breeder is interested in crossing different species and often genera to combine desirable characters. Desirable means which is they like the character. It may be a color or a quality or a quantity to produce commercially superior varieties is called artificial hybridization. Two variety of plants combine together to form the one plant to get the better quality. It is called artificial hybridization. It is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination. Means unwanted pollen grains should not reach in the hybridization. This is achieved by two techniques. It is called emasculation and bagging techniques. Artificial hybridization achieved by two techniques. It is called emasculation and bagging techniques. We'll see what is that. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehiscence using a pair of forceps. This technique is known as emasculation. See, listen, if it is a bisexual flower, removal of the anther from the flower bud stage with the help of the forceps is called emasculation. That is one method of artificial hybridization. The emasculated flowers remove the anther. That flowers, emasculated flowers, have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. To control the unwanted pollination, to cover the emasculated flower, that process is called bagging. When the stigma of bagged flower attains receptivity, mature time, the mature pollen grains collected from anthers of the male parent are dusted on the stigma and the flowers are again rebagged and the fruits allowed to develop. This process is called the artificial hybridization techniques. That is emasculation, bagging, again rebagging. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged means covered before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen. Which pollen we want to, which type of the pollen. Desired pollen and the flower rebagged. Listen, the female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. These are now the pollen grains are ready. Stigma is ready, pollen it is reached. Now the next stage of the flowering plants, the important stage, that it is called the fertilization. In the flowering plants, the fertilization is double fertilization. It's a very important portion. See, listen. After entering one of the synergates, the pollen tube releases the two male gametes into the cytoplasm of the synergate. One of the male gamete moves towards the egg cell and fuses with its nucleus, thus completing the syngamy, fusion of the male and female gametes. This results in the formation of a diploid cell that is called the zygote. Syngamy means fusion of the haploid male and haploid female gametes to form a diploid zygote. This one, after entering the one of the synergates, the pollen tube releases the two male gametes into the cytoplasm of the synergate. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. After the syngamy, the zygote is ready. Next, the other female gamete moves towards the two polar nuclei that is present in the central region and fuses with them to produce a triploid primary endosperm nucleus. That is a one cell but two nucleus is there. So it is a triploid haploid primary endosperm nucleus, as this involves the fusion of 3 haploid nuclei, so it is termed as a triple fusion. Since two types of fusions, one is syngamy and the other one is a triple fusion, takes place in an embryo sac, the phenomenon is termed as double fertilization. Syngamy and triple fusion together, it is called double fertilization. It is an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell and develop into the endosperm, while the zygote develops into an embryo. Thank you.